Hi. So welcome. This is supposed to be the second part of a two-part lecture on linkage mapping. So if you haven't seen the first part yet, then you should be able to find it wherever you found this. And I'd recommend that you look at that first because we went through there the basic principles of genetic linkage. How you can take two polymorphic loci, two markers or a marker and disease locus, work out the phase that they're in, work out whether as they get passed down from one generation to another, whether there's recombination between them or not, calculate the observed number of recombinants, that's the recombination fraction, and then you can calculate the likelihood of observing that number of recombinants given a particular value for the recombination probability, which is to do with how near together or far apart the two loci are, and you construct a statistic called a LOD score, which is the logarithm base 10 of the ratio of the likelihood for observing your data, that number of recombinants, assuming a particular recombination fraction between the loci, against assuming that they're not linked, that the recombination fraction is 0.5. So that's kind of like where we got up to. And what we're going to do now is look at how we can apply that in practice for mapping human disease genes. And the kind of situation we might be in is where we have a pedigree where we have a number of affected children of this couple and this couple, and what this is showing is that both the fathers are first cousins and the mothers are also first cousins. And we would see the disease, which is the people shaded solid, segregating in the pedigree, and we would then genotype some markers, some polymorphic markers, and we would find out the alleles that each person had those markers, and we would try to make some inferences about where the disease gene might be given the pattern of the markers that we see and the pattern of disease that we see. So these are markers going down chromosome 21. There's two microsatellite markers, and there's the APP locus, something in the gene for amyloid precursor protein. And we would take the alleles of the markers, and each marker would have a pair of alleles in each person, and we could use the allele of that marker to say what the parental origin of that allele was. And in fact, it would be which grandparent this allele came from. And we can just color code them. It doesn't really matter what these are. We can color code one allele of this black and the other one white, and then we'll have another one of this locus will code the ones on the same chromosome black and white, so we can get an inherited haplotype that's come down intact through this pedigree, and we'll be able to see the same set of markers going down the chromosomes in these more distant cousins. And then we can see where the haplotype gets inherited intact, where it's black all the way down or white all the way down, and where there has been a recombinant. So here, what we see is that we have, say, the two alleles of these two, which usually go with the other alleles on the chromosome shaded black here, and we see that they've ended up with the alleles which would usually be on one of the chromosomes that's shaded white. So we can say there's been a recombinant here, and we can say there's been a recombinant here. And what we see is, let's see, if we think that the disease variant is on the black haplotype, which seems to be mostly the case, each of these people has got a whole black haplotype that's got the disease. What we can see here is that somebody's got some of that black haplotype but not the rest, and they haven't got the disease. And the inference we can make from that, from intuition, is that the variant must be down here somewhere. It's somewhere on this bit of the black haplotype, because the person inheriting this bit hasn't got the disease. And if we come over here, we see another person who's hasn't got the disease, and he's got the marker at the other end of the black haplotype. He's got this one. So we think that this region here of the black haplotype, if you're calling it that, does not harbor the disease variant. And what we've done now is we've narrowed the disease variant down to an interval which is between this marker and this marker. This gives you this marker, this gives you this marker, and so we can say that we think consistent with linkage would be that the variant causing the disease is somewhere in the interval between the two markers. And that's an intuitive kind of thing, which kind of makes sense. And what we're going to do is kind of formalize that. And we're going to do likelihood calculations, and we're going to use that to get a more sort of clearer grip on what's going on. So we can kind of think about how we will go about linkage analysis in a more systematic kind of way to say a number of things. First of all, we're going to talk about what the genetic model was, and we haven't necessarily said that explicitly, but there we were kind of looking at a situation where, so we look at a situation where we might say, is it a Mendelian disease? Is it dominant? Is it recessive? And then the idea is, if it's a dominant Mendelian disease, if you see somebody who's got the disease, then you know they've got one copy of the disease variant and they've got one copy of the normal gene. And if you see somebody with a recessive disease, then they've got two copies of the disease variant. So understanding the disease model is going to be the start of us making inferences about which alleles at the disease locus somebody has. And in fact, that gets more complicated later because of things like incomplete penetrance and so on. But for now, we'll just take the sort of simple thing where we say it's Mendelian, and we can tell just by looking at somebody's phenotype which alleles they have. So from that, we look at their phenotype, have they got the disease or not, and we will assign the disease genotypes, the genotypes at the disease locus. Then we're going to type some marker alleles, and we're going to work out whether the marker alleles are in phase or not, how they are phased with respect to the disease locus. And that will allow us to look at each meiosis and say, is it a recombinant or is it a non-recombinant? And then we've scored those. We've got a certain number of recombinants, a certain number of non-recombinants, and then we're going to calculate log scores the way that using the formula that we looked at in the previous session. So here, to put that in a more concrete example, here we've got an autosomal dominant disease segregating through the family. Affected, half the children affected, half the children not affected. We're going to say that the penetrance is complete. Well, that means everyone who inherits the disease variant definitely gets the disease. We haven't got anybody who's got the disease variant who haven't got the disease yet, or that they're never going to get it. It's perfectly, fully penetrant. So that's complete penetrance. No heterogeneity or phenocopy. So everybody who's got the disease has got the disease allele. Nobody's got the disease for some other reason. There's not some other gene which is causing the disease in the same pedigree. There's not some environmental thing. There's nothing else. Everybody with a disease has got that variant which causes the disease. And we'll assume it's rare, so there's not loads of them floating around the population. And that means that when we see one, there's only one. We haven't got several disease alleles floating around within this pedigree. We've got one. So then we can start to make some assumptions. So we're going to say, first off, the people who are affected, we know they've got an abnormal gene, an abnormal variant. We'll call that A. We said it's rare, so everyone who's not affected, they've probably got two normal. Well, it's fully penetrant, it's fully penetrant. So everybody who's not affected must have two normal alleles. If they had even one normal allele, abnormal allele, they would be affected because it's fully penetrant. So we know these. Now, we've also said that it's rare. That means if we see somebody with the disease, we think they've got one abnormal allele, but we don't think they've got two because it's super rare and it would be unusual. It's too unlikely. That means we can tell everybody's genotype just by looking to see if they've got the disease or not. Everyone with the disease has got one abnormal and one normal allele, and everybody without the disease has got two normal alleles. So that's fine. Then we can do that for everybody in the pedigree. And we've done that, fine. So then we've got disease genotypes. Now we're going to type them for genetic marker. Here's the genetic marker. We're going to label the alleles of the marker 1 and 2. And we can then follow through the inheritance of these marker alleles, and we can make some inferences about them. So the first inference we're going to make is that the 2 allele has come from this grandfather to this father. And we know that because the grandmother doesn't have a 2 allele, she only has a 1 allele. So we got that inference. We also know that the A, the abnormal allele, has come from the grandfather to the father because they both got the disease. And that means that we know that this haplotype was an A and a 2. And looking at it in terms of biology, we know that the sperm that resulted in this individual must have had an A allele and a 2 allele. Now, we don't yet know whether on the same chromosome or anything or not, but we know that somewhere on all the chromosomes that this sperm was contained, he had the at the disease locus they had the A allele, and at the marker locus they had the 2 allele. And we know similarly that the ovum would have had a normal allele of the disease gene and a 1 of the marker allele, because that's all the mother had, and that's what makes up the rest of this person's genotype. So we're cool with that. So that means from the previous talk that we can say these are in phase. The A and the 2 are in phase, they've been inherited together. That means that we can look at the A and the 2 in each of these children, and we can see whether there's a recombinant or a non-recombinant. We're going to be able to count each of them. So this child has got, and we know this is this 2 allele. We're going to watch this 2 marker allele. This mother hasn't got any 2s, so this is fine. This means it's going to be informative for linkage. She always passes on a 1 for the marker allele and always an N. So we know that we can just look at this father's meiosis in terms of what he passes on to his children. And here we go, we've got an A and a 2, an N and a 1, A and a 2, N and a 1, A and 2, and that all goes through. And there's one person here, where are they, who's got an A and an N, but doesn't have, I thought we, oh, there it is, sorry. One of these people, this child here has got the 2, but they don't have the A. So everyone has either A and 2 or N and 1, except for this daughter who's got 2 and N. So N and 2, she's inherited N and 2 from her father. And so given that these were in phase before, we can say that everyone but her is a non-recombinant and she is a recombinant. So we can mark that down here. This shows it all out. We can label them as non-recombinants and recombinants. And we can now do the sums on those to work out what the LOD score is. So here's our formula for a LOD score for 9 non-recombinants and 1 recombinant. So this is the general formula for the LOD score. We say the recombination probability is theta. We loosely call that the recombination fraction. At any given value for theta, the LOD score will be R, the number of recombinants, times the log of theta, log base 10, plus n minus r times log base 10 of 1 minus theta, minus n, which is here, r plus n minus r. In fact, I see I've jumped notations on you, times log a half. And me jumping notations was that here we use the word nr to mean non-recombinant. Here we mean n to be the total number of recombinants and non-recombinants, and r to be the number of recombinants. So we kind of left that in as a deliberate mistake, because that's the kind of thing you see rattling around textbooks and papers when you try to read them, and it's good to get a heads up about that sort of thing. So we got 1, we put the numbers in there, we've got 1 recombinant, we've got 9 of n minus r, 10 minus 1, 9 non-recombinants, and we got 10 in total, 10 informative children, offspring. And then we can put in a different value of theta into this formula each time to get the LOD score, 0.01, 0.05, 0.1, different values of theta up to 0.5. And at 0.5, the LOD score is always 0, as I told you in the last lecture. And we can graph these numbers out. And what happens when you graph these numbers out is you get a curve that looks like this. And I've got, I guess, 3 points to say about this curve. One is that it's always 0 at 0.5, because that's the null hypothesis of no linkage, and the logarithm of 1, the likelihood ratio logarithm of 1 is 0, so LOD score is 0. It's minus infinity at theta equals 0. So if we say the probability for a recombination to happen is 0, what's the probability for me to observe 1 recombinant and no recombinants, and 9 non-recombinants? The probability for that observation is nothing, 0. It can't happen. The probability is 0. If the probability for recombination to occur is 0, but I do observe a recombinant, then my likelihood for that data is 0, and the logarithm of 0 is minus infinity, so that's gone down to minus infinity there. And then the maximum occurs at this point here, lo and behold, 0.1. So a tenth of the meiosis, tenth of the children are non-recombinants, sorry, a tenth of them are recombinants, 9 are non, 1 is. So the proportion of recombinants is 0.1, and the maximum likelihood estimate of the recombination probability is indeed 0.1. So that's kind of sort of what we'd expect. I'm sure there's massive statistical theory that says why the maximum likelihood estimate tells you or matches the observations, but it does here. So you get a maximum at the place which actually represents the proportion of meiosis which are recombinant. So that was fine. We got it. We got a LOD score curve there, and we can do stuff with that. And we can see that the maximum LOD here is 1.6. And we said before that to demonstrate linkage, you need a LOD score of 3. What you say is this is halfway to showing linkage. If you had 2 pedigrees that look like that, then you would declare linkage. But we haven't got that. We've got stuff that's a bit positive, but looking good, but still nothing to write home about yet. Now, what happens if we didn't have the grandparents? Then we've got 2 possible situations going on. We're still seeing that this person's got the AN allele, A and N, because they got the disease, and the 1 and the 2. But what we don't know is what the phase is. We don't know if the A is going with a 1 and the N is going with a 2, or the A is going with a 2 and the N is going with a 1. And that means that when we count who's a recombinant, who's a non-recombinant, we can count it either way. If the A starts with the 1, then most of these are recombinants, except for this one. But if the A starts with a 2, as in the previous example, then most of the non-recombinants, apart from this one. And what we're going to have to do when we calculate the LOD score is say it's 50-50 either way. We have this more complicated, slightly more complicated function where we say that the likelihood is a half of this one way round and then a half of this the other way around. And of course, we don't know which are R and which are N minus R here. We're just going to label them arbitrarily. So it's half of theta to the 1 plus 1 minus theta to the 9. And this is now a likelihood, not a logarithm of the likelihood. It would look like the other one if we took the logarithm of it. We're gonna add those 2 things together. That's the overall likelihood. We're going to take the logarithm of that, compare it to the logarithm under the null hypothesis, calculate our LOD scores again. And what we see is again that the maximum LOD score is at the same place, 0.1. That's still our best bet because it's either a tenth, whatever. And the data best fit the idea that the recombination fraction is 0.1, but the magnitude of the LOD score has now got less. It was 1.6 and it's now 1.3. So it's come down a little bit. So by and large, phase unknown meiosis are less informative than phase known meiosis, but they still can give you information about linkage. What happens in reality, you just pour these things into a computer program and it works it all out for you and it tells you what the LOD score is. So, and this is what we need a computer program, because when we actually do these studies in real life, we don't have this kind of perfect information. We have some people where you have alleles which are homozygous. We have some people where we haven't got any DNA from them. Got some people where they've done all the diagnosis is they died before they were old enough to get the disease. The mating types happen to be uninformative because you've got 2 heterozygotes together, and you have missing data. And then this is important about the disease model. So let's say we start with a Mendelian disease. You know, if somebody's got the disease, they've definitely got the variant. If they got the variant, they've definitely got the disease. But actually what we had to do was to accommodate some kind of uncertainties. So even if you have a disease which is always penetrant, say Huntington's disease, there's still a question about if you look at somebody of a particular age, 30, you don't know whether they've got Huntington's now, or if they haven't got it now, are they going to get it when they're 40 or 50 or 60? So even Mendelian diseases are not kind of Mendelian in real life, if you like. And what that means is you have to put, instead of the sort of assumption, the logical inference that somebody's got an A and an N, you have to put in a probability function that they might have the A, they might have 2 A's, they might have 2 N's, they might have an A and an N. And that feeds into the likelihood function. That's called the disease model. There's also a question about the allele frequency, which also feeds into the disease model. And basically, you pour all these things into a computer program. You type in the pedigree structure, you type in what every genotype each person has, and then the computer does some very complicated likelihood functions and spits out a LOD score at each of those recombination fractions. So what you would do, you might have a place where you want to look for linkage already. You'd have your pedigree. If you're looking already, then you might say, well, is there linkage there or not? And if there's not, if you get strongly negative loss scores, less than -2, you say, actually, this isn't the gene, forget it, that was wrong. But if you get some somewhat positive results, then you might want to go on and do fine mapping, or you might have to go look all over the genome with a whole bunch of markers, 400 markers to cover the whole genome, find the ones that are positive, get a region that looks linked, and then go in there to do fine mapping and final localization, which I'll show you kind of in a bit. And I've deliberately left this comment just to cheer myself up down the bottom. Recently, if no power, use NGS. And as I said the last time, anyone who thinks that just sequencing a whole genome and the answer just kind of pops out at you is going to be very disappointed when they get when real life confronts them. But there is kind of a point that if you that you might not get very conclusive results from linkage analysis, and if there are some useful looking genes, then sequencing everything, you might prop up some variants that look plausible and which might co-segregate with the disease, and which you might end up saying you think are kind of causative. But it's definitely not a kind of panacea, next generation sequencing. So what do we do in real life? So here is a real Mendelian dominant disease, BPES. I apologize, I always have to look up what this is. This is blepharophimosis, which eyelids, blepharophimosis, ptosis, and epicanthus inversus syndrome. And what that means is that you have kind of drooping eyelids, you have difficulty opening your eyes wide, and you have your epicanthic fold can be folded over the fold over your eyelid. As this is a picture of the normal kind of appearance. And what this does is it segregates as an autosomal dominant disorder through families. And so what this published study showed was a collection of 3 families in which this disorder was segregating, in which linkage analysis was carried out. And obviously, I'm not expecting anybody to be able to read these actual marker names, least of all me. But what you do is you genotype a bunch of markers, 4 markers here, across a particular length of chromosome. You get their alleles, and then you use those alleles. Here, they've actually color-coded the chromosome to reflect how the inheritance of the alleles goes along with the disease. And so, for example, what's a good example to look at? You can see that there's some sort of hashing here, the diagonally hashed chromosome, which is on the right here, which is labeled by these alleles 10, 10, I think 6-1, 10, 10, 6-1, 10, 6-1. You see that these 2 people are affected, and this one have all got that haplotype, which has gone through here. And then if you look over here, you'll see that there is over here, the left-hand chromosome has got exactly the same alleles running down it. So it looks as if this chunk of chromosome has been inherited intact through these number of different meioses, along with the disease mutation that these people who are shaded solid have all got. So that's that's what you see. You see people with the phenotype, and you see some marker alleles, and you can infer the inheritance of the marker alleles. But what we're going to do is put those marker alleles into the linkage program, have it spit out some log scores for us, and this is what we see. So these are different values of the recombination fraction, as we're calling it now, recombination probability of theta. We evaluate the log score at these different values in each pedigree, and each pedigree gives us a log score like this. So you see that this one gives a lot of 1 here, a 1.04, 1.01, and so on, at these different values for theta. Now, as I said in the last lecture, because logs are logarithms of probabilities, and because probabilities multiply, what we can do is we can say that if we add up all the log scores across a pedigree, a particular recombination fraction, that is the same as getting the logarithm of the multiplied up likelihood ratios multiplied across the 3 different pedigrees. So if you multiply them all together and then take the log, that's the same as taking the log and then adding the logs together. So what we do with log scores is we LOD scores is we add them up over families, and then we have a total LOD score. And the total LOD score gives us the overall evidence for linkage, which the three families together provide. And what you see is that for this marker, your total evidence for linkage amounts to a LOD score of 1.4, which is maximal at 0 recombination. So we're not observing anybody's definitely a recombinant. And the information is such that we have a LOD of 1.4. But this other marker, which is a little way down chromosome 3, we actually, the LOD score actually adds up to 2.96 at a recombination fraction of 0. So again, we're not observing any recombinants with either, but this marker, the second marker, is maybe a bit more informative, so it gives a bit higher LOD score. And what you see is that LOD score is as near as, damn it, 3. It's very nearly 3, and this is why it's got published. You know, it's not 3, but it's nearly 3. So this is really looking pretty much as if there's a variant that causes this disorder, BPES, somewhere near this marker. Now, I say somewhere near now while I've got this slide up, but actually it might not be very near. If you look at the LOD is nearly 3 at 0, but if you look out at 1, 2 centimorgans, it's pretty much exactly the same. And even if you look out at 10 centimorgans, a recombination fraction of 0.1, which is a huge distance away in terms of chromosomes and genes and stuff, it's still pretty high. So we're looking at this, we think there's probably something there, but we've no idea if it's here or over here or whatever. Somewhere on chromosome 3, somewhere in this region of chromosome 3. That's what we've got. So that's the kind of thing that happens in practice. Now, if you have a recessive disease, you can do similar kinds of things. So if your disease is Mendelian recessive and you have a couple of affected siblings in your SibShip, then you know that if the parents are unaffected themselves, you know that both of them were carriers. So you know the genotype of the disease locus, here we're saying is big D and little d, where big D is the variant allele which gives you the disease. We know that the people who are affected are both big D, and we can type everybody for a genetic marker. And we can see what genotypes everybody has. What we can't tell from this is, first off, what phase the disease and marker variants were in either of these parents. And the other thing that we can't tell is what disease allele, the variant disease genotypes the unaffected SIBs have. So we know they're not big D, but we don't know if this one's little d, or big D, or little d, big D. So we've no idea, because if they're a carrier, if they have a copy of the variant allele, they're not affected. If they have no copies of the variant allele, they're not affected. So we've got a stack of question marks along here for the disease locus genotypes and the unaffected SIBs. So we only know these and these, and we could do linkage analysis on this, and we do it on those kind of pedigrees if they're a bit sort of bigger, but it's tricky with this kind of pedigree. And you have to think of the two possible phases, and then you could put into the likelihood thing whatever the genotypes could be. But you can do it to an extent. So here is a autosomal recessive form of Parkinson's disease, which causes juvenile Parkinson's disease. So we've got 3 different pedigrees. This one we've only got genotypes from one parent. This one is a product of that first cousin marriage. So these, we think that the same haplotype will have been inherited down this way and down this way, and then it comes together in this person here. So you see that here you've got the 224 and 224 and 108, 108 will both have come from one of these grandparents, and that's called homozygosity mapping. Whereas here you still got the same. These are also what's I going to say? These two are the same, but this one and this one are different. So this one could be a recombinant, and likewise in this pedigree down here. So you know it gets complicated. You observe all these alleles. You don't know what's going on. You put it into the linkage program. The linkage program calculates LOD scores for you. But this pedigree in particular, you can sort of see the general principle that everybody's affected has got the 224 allele and the 108 allele twice, as if they'd inherited 2 copies of the haplotype here. And this person's got 1 copy of that haplotype, and this person's got 1 copy of that haplotype. Now this person has got looks like two copies of like 22418 and 22418, but they're not affected. So did they just get lucky? Are they going to get Parkinson's disease that they haven't got it yet? Or is it the case that Parkinson's disease is nowhere near these variants at all? So you don't have to worry too much about these things. You put those things in, you put in the penetrance, what you think the penetrance values are, and then you let the log score programs calculate log scores, and these are the log scores that they spit out. And here you can see that for that first marker, you've got a log score over 3, and it's maximum at 0 recombination. And for that second marker, if you take that on its own, actually that second marker isn't giving much in the way of evidence for linkage at all, even though it's quite nearby. In one of the pedigrees, there's not much in the way of there's no recombination between the disease and second marker, but in the other one, there's enough, other two pedigrees, there's enough recombinance that overall there's not any overall evidence in favour of linkage, and these log scores are all kind of negative. So you add them all up, and it's just very weakly positive over here, but that doesn't mean anything. But these log scores remain over 3, well out to recombination fraction of sort of 0.09 or 0.1 or so. Right. So then you think, this is cool. I think the gene that's causing this juvenile form of Parkinson's disease is somewhere near this marker. Cool. Now, if you don't get lucky, then what you have to do, as I said, is cover your whole genome with markers. And this would be what we would do, what we came to do routinely. We'd have 400 microsatellite markers, which STR stands for short tandem repeat. So it goes, a diallelic repeat CA, or tetraallelic tetranucleotide repeat CATC, whatever. You have a certain number of repeats, you count the number of repeats, that's your allele number. There's lots of these markers, you spread out over 23 chromosomes, one every 10 centimorgans or so, and you make a map of them across all the 23 chromosomes. You buy this set of markers, and you would genotype them all, and you would find which markers were showing evidence for linkage to your disease, and then you do fine mapping of the string in a bit. And then people nowadays don't use microsatellites because the whole thing has moved over to chip technology to do GWASs. People don't really type microsatellites anymore. So instead, what you can do is use the GWAS SNPs. And it's not 10,000, by the way, it's like 100,000 or 1 million SNPs. And you can, again, they're spread out across all 23 chromosomes, and you use them instead, and you can use them rather than one at a time. There's methods for using groups of them together to get more information from them. And this is what our typical marker panel would look like. You'd order this. These are all the markers down the bottom here. They have different positions, and the position varied slightly according to which company you got it from and where they thought. Obviously, in real life, the position doesn't vary, but where the company thought it came from, they give you slightly different numbers. So these are like different maps from Marshfield or Decode or whatever. And you also know where on the chromosome those were. Heterozygosity was an important thing, is how informative they were, which was, if I genotype somebody at random for that marker, what's the probability that the two alleles that one person has are different from each other? So the heterozygosity property is how probable is it that two random alleles are different, and that's useful because the more probable it is that they'll be different, the more informative for linkage they'll be. If the two markers are the same, they won't be informative. The alleles are the same, they won't be informative for linkage. If they're different, they will be informative, and so that was kind of like useful thing to know about. And the other thing on here is this is the sort of recombination fraction based match. The linkage map is called, and this is the actual physical distance map as measured in DNA basis along the chromosome. So that's what we did, and this was the whole structure of identifying disease genes, and was then used to build up the early drafts of the human genetic map, the human genome in the early stages of the human genome mapping project. Is all based on all of this. So this is what you would do when you're trying to find a disease gene. You type 400 markers. These are a few of them on chromosome 2. They're spread out across chromosome 2, and what you see is that 3 of them are a little bit positive, and 1 of them is just about over 3. And you look at that and you think, oh well, it looks as if the disease gene might be around here somewhere. And this is what we did. Now, the problem that I've alluded to before is that linkage gives a very, very broad localization. So I don't know what's this, is a scale on here? Yeah. So you can see that if I look along the bottom here, This length is about 50 centimorgans, which means that that is about certainly 10 or 20 centimorgans between these two markers. And broadly speaking, you've got a spike, but you've got a spike that's about 5, 10, 20 centimorgans wide. Now that is a huge distance, which will contain, I guess, hundreds of genes to say, oh, we got linkage in that region. So even down to 1 centimorgan, not 10 centimorgans, but 1 centimorgan, which is 1 megabase, you've still got a lot of genes in there to worry about. So how do you get finer? So one thing you want to do is you might try typing more markers, and because it might be that one marker has been uninformative, because it happens to be homozygous in a particularly crucial mating. You type a different marker nearby, it's heterozygous, and then that might tell you, oh, there's a recombinant there. And then you say, oh, that's great. And then every time you get a recombinant, that tells you that the disease locus is the other side of that marker. And so you can you can type more markers and get more recombinants, and try as far as you can to get a minimum shared haplotype, which all the affected subjects have got, in which you think the disease gene might be. But that might not work, because if you think about it, if you suppose you've got 100 informative subjects, right? And then if the recombination fraction is 1%, 0.01, which is equivalent to 1 centimorgan, then you've got 100, they'll mostly be non-recombinants, and one of them might be a recombinant. The expectation is you have 1 recombinant out of 100. But you might be unlucky, and you might not have that. You know, it might just be 100 non-recombinants, or it might be 2 non-recombinants. But what I mean is that out of 100 subjects, you haven't got a great probability. You're not very sure that you're going to get your 1 recombinant. If you do, then you've still only narrowed it down to, you know, if there's 2 markers that are 1 centimorgan apart, one's got a recombinant, the other one hasn't, so it's near the first one, it's near the one that doesn't have a recombinant, you still only narrowed it down to 1 centimorgan. That's a megabase, so that's a lot. So which is why you're going to go on to linkage disequilibrium analysis. So to where linkage disequilibrium analysis comes from is from linkage, and this is one of the key reasons for knowing about linkage. So let's start with the linkage bit. Here you are, you've got your haplotypes, you're narrowing down your haplotypes, you've got a critical region here, you don't think it's there, you're getting smaller and smaller, looks like it might be somewhere between these 2 markers here. But you don't have any more recombinants now, so there's no more information. So now you've got to look at another pedigree, or you've got to find other relatives in the pedigree, more extended relatives, where you're hoping that you'll observe a recombinant. You get further and further away, more and more distant. You start to narrow your region down more and more. You look at the breaks in the haplotypes. You get a kind of critical region which you hope involves fewer and fewer, more and more closely spaced markers. But then you have to go out further and further, and then you arrive in a situation where notionally what you've had is a largish number of generations ago, there was a founder who had a mutation in their disease gene. And then what has happened since then is, in all his descendants, there have been recombinants and been crossovers near that gene, quite near it, very near it, super near it, not very near it at all, which have broken it up. But still, there haven't been enough recombinants to really thoroughly shuffle everything up. So you still end up with a kind of the region of the genome which has got the disease mutation in it and a bunch of variants near it which have got the same alleles as the founder had. And this region can be longer or smaller. So there could have been a recombination crossover here in one of the ancestors of this guy, and a crossover a bit further away in one of the ancestors of this guy, and so on. So the region that's shared around the disease mutation varies a bit between individuals. But the idea is, if you look across a lot of individuals and you genotype them for lots and lots of markers, you will find a tendency for people who are affected with the same disease, because they have inherited from the same founder, to share more than you would expect by chance markers in the region around that gene. And the more markers they share, the closer you are to the disease gene, the more they'll share. So if I come a little bit away here, up here, then 2 of them have got it and 2 of them haven't. Whereas if I come down here, then all 4 of them got it. That's the kind of thing you're looking for when you're doing linkage disequilibrium mapping, and you'll hear more about that in the lecture on association. So linkage disequilibrium, we can define it specifically as a way to the frequencies of alleles at one polymorphic locus are different according to the allele of the locus of interest. So here you have a chromosome bearing a cystic fibrosis mutation, and the frequency of the A allele is 0.9 on that, whereas the background frequency on other chromosomes which don't have that variant is only 0.4. So then you say that the A allele is in linkage disequilibrium with the cystic fibrosis mutation. And here you've got another variant, another polymorphism. We've labeled the others B and B, and here the little b allele is more commonly found on CF chromosomes than it is found on non-CF chromosomes. So the little b allele is associated with cystic fibrosis, and the little b allele is in linkage disequilibrium with the cystic fibrosis variant. And how you apply that in practice is you genotype a lot of very, very tightly spaced markers along your genome, and you measure your linkage disequilibrium. This is a measure of linkage disequilibrium, which you'll be told about with some measure, just that's what it is. It goes from 0 to 1. And you try to find where is the linkage disequilibrium maximal. So this, we graphed a number of different markers along here. And this is what this, by the way, you'll see the same sort of diagram when you look at a close-up of a genome-wide association study. People will see exactly the same thing, except it will be a p-value up here rather than LD measure, the log minus log p of a p-value. Just to point out how I want to just point out how closely spaced these markers are. So before we talked about 1 centimorgan, 1 megabase. Okay, so now this is measured in kilobases. So 1,000 kilobases is 1 megabase. So this here, this distance is 1 megabase, 1 centimorgan, and that is where, that's kind of like the absolute limit of where we might hope to get our resolution in terms of linkage studies. And there will be a whole bunch of genes within this region. Then we go on to use markers which are much more closely spaced than 1 centimorgan, and you'll see that I don't know how many are. I've got on the x-axis, but there's a bunch of them. And then we can see that there is a place where the linkage disequilibrium is maximal between our markers and the cystic fibrosis mutation, and we can use that linkage disequilibrium to actually identify, and this is published in Science, this is where the cystic fibrosis gene is. We can map it using linkage disequilibrium. So that is how we used to do stuff, honestly. It was a very, do linkage, get an approximation, do fine mapping, trying to detect association, linkage disequilibrium, and then go looking in there for genes that look like they might be doing something and try to find a variant in those genes. So a couple of things about the comparison between them. So linkage disequilibrium association is observed as association of an allele with a phenotype, and it's that the 2 alleles are going together in the level of population. The 2 alleles tend to be found together. There's a particular marker allele which tends to be found more often than by chance on the chromosome bearing the disease mutation. Linkage is not about alleles, it's about 2 loci. You say this locus is linked with that locus, but if you look at different pedigrees, it will be different markers co-segregating with the disease in different pedigrees, different marker alleles. So you talk about a relationship between the loci in linkage and the alleles in linkage disequilibrium. And in a way, the alleles, for linkage disequilibrium to be detectable, it has to be the same allele which is associated in a bunch of unrelated subjects. I'd say 1,000 people with cystic fibrosis, and they've all got to have, on average, it's going to be A allele that is going to be associated, the A marker allele, with the disease. And there is a problem with that, which you will hear about, but just kind of say, supposing, as is true, there's more than one different mutation that will give you cystic fibrosis. Now, with cystic fibrosis, they were lucky because there was one that was especially common. But supposing you're in a situation where you have a number of different variants, each of one of which, if they damage that gene, will give you the disease. And if those different variants are all segregating in your population, then any particular marker allele might be associated with one of those variants, but it's not going to be associated with the others. It will be random with respect to the others, maybe, or whatever. And so if you have allelic heterogeneity of the disease locus, that makes it very difficult to detect linkage disequilibrium, which is kind of one of the reasons why in practice a lot of association signals tend to be quite weak. And in a way, people were quite lucky with cystic fibrosis that it was a less challenging problem than it needed to be. Okay, so there's some references on the last slide. These slides are going to be available from wherever you got this this video from. I'll kind of just finish up there. I think that to summarize really the things about linkage, I think it's very important historically to get an idea of how we got to where we are now with our understanding of being able to, you know, just have a whole sequence of every human chromosome. It was very productive also in terms of identifying some diseases. When you hear about, for example, Huntington's disease. Huntington's disease was identified, the gene for that was found many years ago using these techniques. So they're not used so much now, but I can tell you that it's taken from the time that that Huntington's gene was identified many years ago for us to be starting to get treatments for Huntington's disease. So it was very helpful that these early methods were successful for actually, you know, a very large number of Mendelian diseases. And the other thing I think to have got from understanding linkage analysis is it's the first brush with, for some people, with likelihood ratio testing. So it's not like testing p-values, it's like you're comparing an alternative hypothesis to the null hypothesis. You take the likelihoods of those two hypotheses, you take their ratio, you take the logarithm of that, and that log of a likelihood ratio is a way that you're saying how much support is there for a particular alternative hypothesis. And in this case, the specific alternative hypothesis is that there is linkage, that the recombination fraction is less than 50%. Okay, that's, I guess, what I wanted to tell you about linkage. I'm going to finish up there, and I'm going to wish you the best of luck with the rest of your studies. Bye-bye.